Hello everyone, welcome to today's live broadcast, Spotlights on T-Cells, New Developments for Translational and Basic T-Cell Research. I'm Julie Clark and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by Miltenyi. The Miltenyi Biotech mission is to improve scientific understanding and medical progress by providing products and services that advance biomedical research and cellular therapy. Honoring this mission drives our commitment to support the translation of basic research into therapy in the areas of immunology, cancer, neuroscience, and stem cell biology. For more information, please visit www.miltenyibiotech.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speaker, Dr. Anne Richter. Dr. Richter studied biology at the University of Cologne, Germany. She obtained her PhD working on T helper cell polarization at the German Rheumatism Research Center in 2000. Afterwards, she joined Miltenyi Biotech as a project leader in R&D reagent department, developing methods and reagents for cell separation, culturing, and analysis. To date, she heads the T-cell immunology and flow analysis group, focusing on the development of innovative tools for research applications in the field of adaptive T-cell therapy. I'll now turn it over to Dr. Richter.
Hello and welcome to this seminar today. Thank you, Julie, for the introduction. Today, I would like to guide you through several topics. The first is I'd like to give you a short introduction into Miltenyi Biotech. Then I will come to discuss with you T-cell workflows. And the last point, I will focus on the flow cytometric analysis for sensitive and rare cell analysis. So first of all, some words to Miltenyi Biotech. We are a company with subsidiaries in 25 countries, and we have distributions in another 34 countries. Our headquarters is located in Germany, Bergisch Gladbach. That's a small town close to Cologne. Here, our research and development work, as well as production, is done, and especially the manufacturing of our GMP and clinical grade products that's performed at a second site in Germany. It's located in East Germany. So our mission is to serve the needs in life science research and to translate new scientific findings into clinical applications. We are actively developing reagents, media, buffer, instruments plus software for applications in the field of sample preparation, magnetic cell isolation, flow cytometric analysis, cell culture, designed for basic research purposes. Important is to know that we build up our clinical product portfolio with the same technologies as we use them for our research product. This fact extremely simplifies the translation of applications from research level into clinical practice. We are closely working together with investigators in early phases of development of clinical applications to ensure the most easiest and fastest translational process. In many cases, even the clinical applications are the driver to choose and implement the technology into our company, which we then additionally provide in our research portfolio. Later on, I will mention some examples. But first, I'd like to start with our max cell separation technology. And this is a business Miltenyi started with more than 25 years ago. And we introduced the max MicroBead technology. This technology uses nano-sized paramagnetic beads in combination with columns and magnetic separators for either positive enrichment of cells, depletion of unwanted cells, or isolation of untouched cells. This system was developed to enable cell isolation without affecting the cell. Besides, we offer a magnetic cell separation system, Max Express, which consists of micro-sized beads and a separator. The system does not need a column and is especially useful for fast depletion of unwanted cells, for example from whole blood. But let's have a closer look to the MicroBead technology. Why do we utilize a column? In general, the magnetic separators in column-based and column-free system have a comparable magnetic force. The difference between the system is the column. So when the column is placed into a mag separator, the gradient of the magnetic forces gets amplified by 10,000-fold due to the little iron spheres that are packed in the column. Thus, a minimal labeling of cells with tiny nano beads is sufficient to retain labeled cells in the column. The iron spheres have a cell-friendly coating. Moreover, cells can run freely through the column and can be washed thoroughly to remove unlabeled cells, cell debris or contaminants. All this contributes to the fact that MAX columns allow for cell separation with minimal effects on cells. Besides this, MAX cell separation systems are very easy and handy, as it will be shown on the next slide. So basically, the magnetic separation using the MAX MicroBead technology has 3 steps. The first step is the magnetic labeling of cells in suspension. The magnetic nanobeads are coated with antibodies which recognize certain cell surface antigens on cells. And secondly, then the cell suspension with magnetically labeled cells is put onto a column which is fixed in a magnetic separator. The magnetic forces of the separator, which are amplified via the column, will retain now the labeled cells in the column. The unlabeled cells flow through the column and can be collected as depleted cell fraction. In a third step, the cells retained in the column can simply be eluted from the column, which is then outside the magnet using a stamp. And this fraction is then the magnetically labeled cells, the positively enriched cells. The MicroBead technology is the gold standard in magnetic cell separation. It is used for basic research applications, and furthermore, it's used for various clinical applications, also for positive enrichment of cells and subsequent injection into patients. So we are offering a comprehensive portfolio of MicroBead-based products for separation of many cells, but as shown on this slide, we have already a huge portfolio to separate mouse and human cells, pan T cells, as well as subsets of T cells like naive or memory T cells or T regulatory cells, or even cytokine-expressing cells. In the following, I'd like to share with you our work to streamline research on T cells, showing 2 examples of workflows. The first one is the workflow for mouse T helper cell differentiation in vitro. This slide shows to you a simplified model of differentiation of naive T helper cells into cell subsets having various different effector functions. Differentiation of naive T helper cells starts with its primary activation via the T cell receptor. In vivo, the T cell receptor recognizes peptide antigens presented on MHC class II molecules, which are on the cell surface of professional antigen-presenting cells like dendritic cells. But in addition to this first signal, the TCR triggering, co-stimulatory signals are required to ensure the full activation of the T cells. For example, CD28 on the T cell has to be triggered. Additional signals delivered by cytokines during the primary activation phase imprint a certain differentiation program in the T helper cell. For example, the presence of IL-12 during the activation instructs the T helper cell to develop in a Th1 cell. A milieu of TGF-beta, IL-6, IL-21, and IL-23 will induce the differentiation of Th7 T cells, whereas IL-4 induces Th2 cells. So, and several more T helper cell subsets are described, such as Th9 cells, induced regulatory T cells, or T follicular cells. The hallmark of each of the T cell subsets is a certain set of expressed transcription factors which direct the expression of a certain effector cytokine pattern. T helper cell 1 cells are characterized by the expression of the transcription factor STAT4 and TBET, which are inducing the expression of gamma interferon and TNF-alpha. IL-17 cells are marked by the presence of STAT3 and raw gamma T, which are required to induce IL-17 and IL-22 expression. The high degree of the different effector cytokines is important to fight appropriately against the different pathogens we are attacking by, or to maintain tolerance towards harmless antigens. And also inadequate effector functions can result in diseases. And this is the reason why researchers are interested to understand the differentiation process and to analyze T helper cell effector patterns. The T helper cell differentiation process can be mimicked in vivo, and that's most easiest with mouse T helper cells. So, for this approach, several steps are required. First, mouse lymphoid cells need to be prepared, for example, from spleen or from lymph nodes. Then the naive T helper cells have to be selected and in vitro cultured. Reagents for activation of the T cell receptor and co-stimulatory molecules are provided, as well as cytokines to induce the differentiation pathway, as well as cell culture media is needed. So, and to prove the differentiation, the analysis of, for example, transcription factors is done, or effector cytokines. So, coming to the separation of naive T cells. So, naive T cells are characterized by the expression of CD62L and the absence of the memory marker CD44. Based on this marker, we provide magnetic separation kits to isolate these cells. For example, as shown in this slide, we isolate naive T cells using the naive CD4 T cell isolation kit. This MicroBead-based kit depletes all non-CD4 T cells and additionally CD25 positive Tregs and effector cells, as well as CD44 memory cells from a lymphoid cell suspension. On the left-hand side, a flow cytometric analysis of cells before the magnetic depletion is shown, and in the middle and on the right side, the depleted cell fraction. So, as depicted in the upper row, you see the purity of the enriched CD4 positive T cells, and in the middle, you see the purity of the naive T cells isolated. And you can easily see that the CD44 negative, CD62L positive cells are enriched to a purity of more than 96 and 97%. In this example, we show a manual enrichment of the naive T cells as well as an automated enrichment using the Automax Pro instrument. And the lower row shows you that also the Tregs and effector cells are gone using this depletion kit. Of course, we were interested to look if the depletion really resides in naive T cells. And one of the characteristics of naive T cells is that they do not express effector cytokines. And therefore, we did a short-term stimulation of either the unselected spleen cells or selected naive T cells with p-immunomycin. And afterwards, we did an intracellular cytokine staining for gamma interferon. And as you can easily see, whereas in the spleen cells, gamma interferon producing cells are detectable, within the naive T cell compartment, no gamma interferon producing cell is depicted. Having now isolated the naive T cells, these T cells need to be cultured. For in vitro T cell differentiation, several reagents are required. Cell culture media, activation reagents, cytokines to induce the effector cytokine expression. And cell culture is very complex because many reagents are provided to these cells which have an effect on these cells, either wanted effects, but also undesired effects can be caused by undefined components and ingredients. Especially driven by our clinical applications, we were interested to develop reagents and media which are defined. First of all, from regulatory perspective, clinical grade products require to be defined, standardized. And defined and standardized reagents are key elements to enable reproducibility in your application. But that's true not only for clinical application, but it's also of huge advantage for research application. So for this differentiation process, you will need cell culture media. And for this purpose, we developed special media to culture T cells, the Tex-Mex medium. For T cell activation, you have several possibilities. Either you can polyclonally stimulate cells using soluble and plate-bound antibodies, recognizing and binding to CD3 and CD28. We developed artificial antigen-presenting cells. These are based on big beads, the MicroBeads, and these MicroBeads are coated with agonistic antibodies towards CD3 and CD28. For antigen-specific activation, we use pools of peptides which cover the complete protein sequence of an antigen, and these pools allow the activation of CD4 as well as CD8 positive cells. So to induce the differentiation of certain subsets of effector T helper cells, inducing cytokines is needed. And for this purpose, we developed recombinant cytokines, and these are animal and human-derived component-free cytokines to avoid undefined side effects, which is important for clinical, but also for research application. And last but not least, we are using for our differentiation process also antibodies to neutralize inducing cytokines to inhibit the wrong differentiation program. Just to mention what we tested when we developed our Tex-Mex medium. This medium is not only used for polyclonal activation of pan T cells, but also used for antigen-specific T cell activation. Also regulatory T cell expansion is done with this medium, and we use it also for mouse T helper cell differentiation. Beside the medium, you need for your differentiation program T cell activation reagents. So in this slide, I'd like to show you an example where we use the artificial antigen-presenting cells, meaning the MicroBeads loaded with CD3 and CD28 antibody for the activation of mouse T cells. For this experiment, we labeled spleen cells with CFSE to follow their proliferation and activated them with the artificial antigen-presenting cells. And as you can see already on day 1, you see a nice expression of the activation marker CD25, which is upregulated in the CD4 compartment. And on day 2 and day 4, you clearly can see that the CFSE dilution CFSE labeling declines, meaning that the cells are proliferating. And when we determined the expansion rate after 8 days, we found out that CD4 as well as CD8 T cells proliferated by up to 100-fold. We use the artificial antigen, the artificial APCs not only for expansion of mouse T cells, but it's also used for human T cell activation expansion, for human Treg expansion, and even we are designed a Treg suppression assay using this the MicroBeads. So beside the cell culture medium and the activation reagents, you need cytokines to induce the differentiation pathway. We bundled the required cytokines as well as the blocking antibodies into a kit that we call CytoBox. This slide shows the result of a 5-day differentiation of max isolated naive CD4 T cells, which are stimulated with CD3, CD28 MicroBeads in the Tex-Mex medium, and in addition with either reagents to induce Th1 differentiation, Th2 differentiation, or Th70 differentiation. We prove that the differentiation works by staining the cells for masked transcription factors, and as you can see on the left-hand side, Th1 cell cultures, in these cultures, 99% of all cells are positive for Tbet. Within the Th2 culture, 99% of the cells express GATA3, but not Tbet is expressed, and most of the cells within the Th1, Th70 culture express the key master transcription factor rho gamma T. Besides the analysis of master transcription factors I showed you before, we also analyze cells for the relevant effector cytokine expression. And after reactivation with PMA-Unimycin on day 4 of these differentiation cultures, we detected in Th1 cells, as expected, gamma interferon, but no IL-4 expression. In Th2 culture, IL-4 but not gamma interferon production was detectable, and in Th17 culture, IL-17 producing cells but not gamma interferon expression was detectable. Interestingly, we observed when we compared the culture medium RPMI and Tex-Mex that the level of the cytokine expression was much higher when we used the Tex-Mex medium. All in all, this means that we have established with all reagents for cell separation, cell culturing, and cell analysis a workflow for the in vitro differentiation of mouse T helper cells. But what's about to identify and characterize in vivo differentiated T helper cells? For this purpose, we established the cytokine secretion assay technology. This technology is unique and allows the detection as well as the isolation of viable cytokine-producing cells. The principle of the cytokine secretion technology is as follows. After T-cell stimulation, cells are labeled with a catch matrix. This catch matrix consists of 2 antibodies conjugated to each other. One antibody recognizes CD45 antigen on all leukocytes, and a second antibody is specific for a cytokine, for example, IL-17. The catch reagent is anchored to the cell surface via the CD45 antibody. When cells secrete the cytokine, this cytokine is catched by the catch matrix on the secreting cell. The cytokine-producing cell can afterwards be fluorescently labeled with a second anti-cytokine antibody, which allow flow analysis or even flow sorting. Alternatively, the cytokine-secreting cells can be magnetically isolated after an additional MicroBead labeling step. This technique is available for many cytokines, and one example I like to share with you is the detection and the magnetic isolation of IL-17 secreting cells from mouse spleen cells. We detected in the simulated sample 0.36% of IL-17 producing cells among the CD4 positive T cell population, and after the magnetic enrichment, we achieved a purity of 60%. Besides CD4 cells, also other cells in mouse spleen cells are able to produce IL-17, and they are even also enriched. The same technology allows also the isolation of gamma interferon secreting cells for manufacturing of clinical grade virus-specific T cells. This procedure run in a fully automated way using a cell processor, the Clinimax Prodigy instrument, which is shown here in the picture. To summarize this part of the presentation, so we established a whole workflow for in vitro differentiation of CD4 T cells, starting with the magnetic isolation of naive cells, proceeding with the cell culturing, and finishing with analysis of the differentiated cultures. In the next slides, I'd like to discuss a workflow for research with human regulatory T cells. Human Tregs are mostly isolated from blood products like PMMC or whole blood. They are enriched according to the phenotypic cell surface markers CD4, CD25, and CD127. Tregs are expanded and are characterized afterwards, including the detection of, for example, FOXP3, the key transcription factor, or other phenotypic markers are analyzed, or even the Tregs are functionally analyzed using a suppression assay. To allow a very fast enrichment of human Tregs directly from whole blood within just 30 minutes, we combine both of our MAX separation technologies, the MAX Express and the MicroBead technology. The protocol starts with incubation of whole blood cells with a cocktail of magnetic MAX Express beads to label non-target cells, but also of CD25 positive cells with MAX MicroBeads. After a short incubation step, the sample is placed into a MAX express separator to deplete unwanted cells and red blood cells. The supernatant is collected and directly transferred onto a column which is placed into a MicroBead magnet. CD25 positive cells are enriched in the column, unwanted cells flow through. The target cells, namely the CD4 positive, CD25 positive, CD127 low cells, are then eluded from the column. What is the result of this enrichment? With the flow cytometric analysis, we determine the purity and the yield of the isolated Tregs. Overall, we achieved a 90% purity of CD4, CD25 positive cells and a 75% purity of CD4, CD25 positive, CD127 low cells. Importantly, the number of isolated Tregs is 2.5-fold higher compared to an isolation strategy which first needs the separation of PBMC via a density gradient centrifugation step. These isolated cells can also be expanded using the artificial APCs, which specifically were developed for expansion of regulatory T cells. Here an example is shown where we use also the Tex-Mex medium, IL-2 and rapamycin. And we detected that the cells expand by a factor of 40-fold within 3 weeks. The frequency of FOXP3, so the key transcription factor which is specific for Tregs, remains stably expressed during the whole culture, in this example around 70%. In summary, I presented to you a workflow for research on regulatory T cells isolated directly from whole blood using 2 max separation technologies, doing expansion of these cells with our activation tools, cytokines and Tex-Mex medium, and doing flow analysis to phenotype the Tregs. We also established a Treg suppression assay to test the functional capacity of the Tregs, but I have no slides today for this. And what is also important is that we, beside a portfolio of reagent and technology that we established for research application with Tregs, we provide also whole workflows for GMP manufacturing of Tregs, including the magnetic enrichment of cells, expansion of these, as well as a quality control where we assess the phenotypical and functional properties of the cellular products. The last topic I'd like to address is the flow cytometric analysis of rare cell populations. What is important to allow sensitive and rare cell detection by flow cytometry? For example, the ex vivo detection of antigen-specific T cells. One thing which is very important is that the flow cytometer has to have a very low carryover of cells. Secondly, the number of cells that has to be acquired have sufficient high enough to be able to detect these rare cells. But even the analysis of large samples on the flow cytometer has its limit, because low-frequent populations cannot be discriminated from other non-target cells, as the large number of non-target cell signals overlays with the signal of the target population. Here, the magnetic pre-enrichment of target cells prior to the flow analysis is a solution. Furthermore, a bright staining of the target cell population is necessary. And a specific labeling of the target cells is also required. I'd like to address 3 of these points in more detail: the pre-enrichment, the bright staining, and the specific labeling. And I will start with the topic of magnetic pre-enrichment. To allow a very high solution characterization of rare CD4 positive antigen-specific T cells, we developed a method we named Rapid Antigen Reactive T Cell Enrichment, Rapid ARTE. And this method allows the characterization of rare cells within 1 day. and it's really a fast alternative to long-term culture with repetitive antigenic stimulations to allow for detection and characterization of rare antigen-specific T cells. So the protocol starts with a short-term antigenic stimulation of PBMC for several hours, for example 3 to 6 hours, with an antigenic peptide. After this period, within the cell culture dishes, we add antibodies for cell surface markers like CD4 and CD3, or even for characterization of memory and naive cells. And we are doing as well magnetic labeling of the antigen-activated T cells using the activation marker CD154. So we do a direct labeling of CD154 with MicroBeads. And directly after this labeling step, cells are fixed. All these steps are done without any washing or centrifugation step in the culture dish. After having fixed the cells, these are loaded onto the column. The target cells, meaning the activated CD154 positive T cells, are retained on the column via the magnetic labeling. To analyze now the cytokine repertoire within these antigen-activated CD4 positive T cells, an intracellular cytokine staining is performed on the column. And after elution of the target cells from the column, the flow cytometric analysis can be done on the MaxQuant instrument, and that's run automatically, so this analysis can be performed overnight. The whole enrichment and labeling procedure takes in total just 1 hour. No centrifugation is required. And when we calculate additional 6-hour stimulation period, it's clear that this protocol can easily be performed within a working day. We use this method to characterize, for example, the repertoire of fungus-specific T cells. In this example shown here, we stimulated PBMC from a healthy donor with Aspergillus lysate for 7 hours. Afterwards, the Aspergillus-specific CD154 positive cells were magnetically enriched and intracellular stained for various cytokines. In healthy individuals, normally around 0.1% of the CD4 T cell repertoire reacts towards Aspergillus. Using now the magnetic separation of CD154 positive cells, we enriched the Aspergillus-specific T cells here to around 30%. When we now screen for the effector cytokine repertoire within this population, we clearly detect gamma interferon producing cells. In this example, 4.2% and only 0.6% are IL-10 producing cells. In comparison, without the enrichment before the flow cytometric analysis, we detected 0.05% of gamma interferon producing cells. So the gamma interferon producing cells were overestimated, whereas IL-10 producing cells could not be detected because of their low frequency. By this method, now small population within a complete T cell repertoire of Aspergillus and Candida-specific T cells were identified, and you can clearly see here the differences in the cytokine repertoire between T cells specific for one or the other of these fungus. For example, IL-17 and IL-22 producing cells are much higher in frequency within the Candida-specific repertoire compared to the Aspergillus-specific repertoire. These differences would not be uncovered without this method. And next, I'd like to discuss the importance of a specific labeling. You may have noticed the publication by Andrew Badbury and Andreas Plückthun about the need to standardize antibody reagents for research purposes, for example, by using recombinantly engineered antibodies. Already before this comment came out, we implemented the manufacturing of recombinant engineered antibodies into our company. Our reaffinity technology selects the best clones with the highest specificity and affinity. They are engineered not to bind to Fc receptors. For example, FCR blocking is not required to inhibit background staining or false positive events. They are optimized in sequence. That means we eliminated unwanted conjugation sites within the variable regions, which result then in a loss of antigen binding. And all our rare antibodies are of human IgG isotype and kappa light chain. So that means that only one isotype control antibody is required for the analysis. In sum, this means that recombinant engineered antibodies allow a standardized manufacturing process, which results also in optimal performance of these reagents. Last but not least, the bright staining is needed. We are developing new fluorescent dyes which extend the brightness of the conventional dyes. For example, the new dye BioBright Fits, conjugated to CD4 or CD8 antibodies, results in higher mean fluorescence intensity than a simple Fits conjugate, but it is as bright as a PE conjugate. Brightness matters in multi-parameter analysis with dimly expressed markers, for example, CD25 to identify Tregs. With new brighter dyes, the flexibility for selection of antibody reagents is much higher. And in this example, the characterization of human Tregs is shown, which were isolated from PBMC. And on the left-hand side, you see the gating of viable CD4 positive T cells that were identified by excluding viability 420 fixable dye. And we use CD4 APC Vio 770 to identify the CD4 cells. Then T cells were also stained for CD25 BioBright Fits, and we exclude CD127 positive cells. The Tregs were furthermore characterized and subdivided in memory and naive population using CD45 RO Vio Blue and CD45 PE Vio 615 conjugates. And for both of these compartments, we then looked for FOXP3 expression, and we looked for proliferating and recently activated cells using the intranuclear marker Ki67. So the combination of new bright biodyes with the rare affinity antibody creates really new standards in multi-parameter flow analysis panels. To sum up my talk, for our T-cell workflows, we have established optimized tools for sample dissociation, sample cell separation, cell culturing, as well as for cell analysis. And we are not only providing this tool for research purposes, but we are also setting up these tools for clinical application. With this, I'd like to thank you for your attention, and I hope for a very fruitful discussion. Thank you.
Thank you, Anne, for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. All right, so our first question is, did you ever test the functionality of your isolated FOXP3 Treg cells?
Yes, that's a very good question. As I mentioned, we have developed extra reagents to set up a suppression assay. And this assay uses also the MicroBeads coded with CD3 and CD28 antibodies. And we nicely can see that the Tregs we are isolating are suppressing. We usually use a flow cytometric assay where we label the responder cells with a dye, and we looked at the dilution of this dye to define the suppressive activity. And for all our separation reagents, we did this, and this clearly shows that the Tregs are functional after the magnetic isolation as well as after the expansion phase.
Thank you, Anne. We have another question. We always use plate-bound antibodies for T-cell activation. Do your activation beads stimulate more efficiently, and do I need to remove them afterwards?
Yeah, we had a lot of house experience with plate-bound antibodies for T-cell activation, and our experience was that there were not really reliable protocols available, and we had problems to reproduce our results. And with our MicroBeads, we were able to standardize the activation of the cells. So, and in general, these beads have not to be removed from the culture during the expansion phase.
Thank you, Anne. The next question is, what temperature do you store your sample reagents?
So when the reagents are delivered to the customer, most of the reagents are stored within a fridge. And if this is not the case, then we mark it on our reagent how to store the reagent.
Thank you, Anne. The next question is, do you have a comparison between using CD3, CD28-based activation T cells versus max IBeads?
Yeah, as I mentioned before, we did this in-house, and we had problems to reproduce the results with CD3, CD28, and therefore we used the MicroBeads.
The next question is the TexMax media a ready-to-use medium for T cells, or do I need to add supplements?
Yes, that's an important question, because normally you add for the media serum, and serum is a very variable component, and this medium works without any addition of supplements. So you can use just this medium for cultivation of your T cells.
Thank you. Will the slides be available online?
Yes, there will be online available.
All right. After stimulation with MicroBeads, do you need to remove beads prior to analysis by flow cytometry?
So the big beads, after expanding of the T cells, the beads are not any longer sticking to the cells, so they can be discriminated from the cells.
Thank you. Let's see. Can you give us direct link to site for reagents? And what is the average cost for the media with supplements?
I'm sorry, Julie, can you repeat, please, the question?
Yes, this individual would like, I guess, more information as to how to find the reagents directly, and I believe that we will be providing information on the Miltenyi Biotech link archived with this presentation.
So I think you will find information for our reagents on our website.
Thank you, Anne. The next question is, new dye you used did not increase the number of cells, only increased MFI. Did you use these dyes on any low MFI antigens?
Yeah, that's also an important question. I showed here an example with VioBright fits for CD4 and CD8 T cells, and these markers are highly expressed, and normally you do not have problems to detect them. But I showed one example where we used the VioBright fits to identify Treg cells, and also there we achieved the same mean fluorescence intensity than with a PE conjugate. So that means that you have now more options also to stain these dim markers like CD24, CD25 with other markers, and you have the PE and the APG channel free for other dim markers.
Thank you, Anne. After isolation, do you see death of some of the Tregs? If so, what percentage of Tregs die before they start expanding?
So we are looking carefully to our culture, and we normally do not detect dying Tregs.
All right. How do you compare the permanent perm stain on columns with the conventional perm fix method?
So this can easily be done because you can, after separation of the magnetically labeled cells, elute the cells and do the staining then either in suspension, or you leave the enriched cells on the column and perform then the intracellular staining. And afterwards you can go to the flow cytometer and compare the performance of both of these protocols, and it will result in the same.
Thank you, Anne. Did you ever test the functionality of your isolated FOXP3 positive Treg cells?
Yeah, I think we had the question before, but so yes, we normally test the functionality of our Tregs after their isolation and also after their expansion. And for this, we designed an extra assay, a functional assay, the Treg suppression inspector. And this allows you to test the functionality of the Treg cells.
Do you have MicroBeads expressing only CD3 and only CD28 antibodies? I need to compare stimulations by each antibody individually.
Yeah, so we have predefined kits where CD3 and CD28 is already provided in the kit, but we also provide anti-biotin MicroBeads, and you can have also CD3 and CD28 biotin antibodies separately to use this in a flexible way and to design your artificial APCs as you want. Yeah, you can even use further antibiotin antibodies and load these MicroBeads to test their influence.
All right. If there are no more questions, I'd like to thank Anne Richter for her presentation. Anne, do you have any final comments?
So I'd like to thank you all to the audience, and yeah, please feel free if you have further questions to contact one of us at Miltony, either go via the tech support. So we will be happy to take your questions and to help you.
Thank you, Anne, for your presentation again, and thank you, Miltony Biotech, for making today's webcast possible. Today's webcast will be available for on-demand viewing through December 23rd, 2016. You will receive an email from Labroots alerting when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.